In this video, we're going to show you how to work up in Excel your area of lipid droplets data. And so what you want to do first is, whether you copied it or you saved these data from the actual images, you want to set it up so it's well labeled. And I have to emphasize that, right? So that you know what you're doing, because I've seen a lot of students in the past where they won't label something correctly, or they'll have two different people working on it or three different people working on it, and they all decide to label things differently or not label, and then data's been mixed up, and then they're a whole week behind because they have to redo all their data. So make sure you're labeling things well. For example, here, they have a category for each of their conditions, a column, and then in this case, because you're dealing with area, it doesn't really matter which cell has which, specific spots, because it's just average area is what we're interested in. And so they have their data here in their column, and it goes all the way down here for several ways. And notice they have different numbers for each of those. I always encourage students to have the same number of samples if possible. It just makes it a little bit prettier. And really, you're going to have so many samples, in this case, 132 samples. And so, you might as well get them as even as possible. And so if that means going back to that image and counting a couple more spots, so be it, right? So that's something I would encourage you to do, because otherwise, when you go to compare the numbers, it comes out different. So I've already done all of these here just so you have them here, but I'm going to show you how I got these answers here. So the first thing you want to do is, for each of the categories, and your categories are going to be different, and you may only have 2 categories, you may have 3 categories, or you may have 6 categories, depending upon your experiment. But let's say, they're listed in this lauric acid here, this fatty acid condition, and we wanted to calculate the average area of the lipid droplet. So that's the mean, all right? So we're calculating the mean in what we call arbitrary units, or A.U, arbitrary units. So you can have that in your chart here in a little bit. So I'm losing that so you can have this all on the same screen. But to calculate that, it's pretty straightforward how you're going to get that. So we're going to calculate that as average. So how you can do that is a couple ways. You can click on this insert function, or you can hit equal sign. Both of those do the same thing. It just kind of is different ways to get to the same thing. Here, average is one of the samples that's coming up, and you can do that, and you can highlight the samples here. There's all kinds of ways that you can do this. Type in equal sign, click on that, double click average. It's doing the same thing. And then what you would do is then you were going to highlight all the samples in that category. And then once you've highlighted all the samples, you can hit enter, and then that should bring up the average here, okay? And then to calculate the standard deviation, what you're going to do is same thing. You hit equal sign, and you're going to type in STD. And I always use, there's lots of standard deviation function types. The one that's the go-to for me is the STDEV, this one right here. It's very common. So what we're doing is we're calculating the standard deviation, or the variation of your data. And so then we would go down here and highlight this entire section again. Hit enter. Okay. And now for standard error doesn't have a set formula in Excel. And so I'm going to show you how to calculate standard error. So how you calculate standard error here is this formula. You're going to take the standard deviation divided by the square root of the number of samples. So here's how we're going to calculate it. Equal sign, standard deviation, which is what we just calculated up here, divided by the square root of what's here. Now to get square root, it's SQRT. So square root, and then we can just click on number of samples, and then you have to close parentheses here. All right, so that's important. Otherwise, it's going to give you an error, and it'll try to correct it most likely. But so what it was equal sign, the standard deviation, which you just calculated, divided by SQRT, parentheses, the number of samples, close parentheses, and then hit enter, and that would be your standard error. And notice both these standard deviation and standard error are plus minus the mean, right? And so our standard deviation of the mean here is our average is about 17 plus minus 9. And the standard error of the mean is 17 plus minus 0.8 roughly. And so all this data here is important. In our graph, we're going to graph the average and the plus minus standard error, and we're also going to indicate the number of lipid droplets that we counted in our calculation. And then you would just repeat it for these other two samples here. So I'm going to go ahead and delete those out, and so then you would just repeat it over here. So equal average. In this case, you would go to your oleic acid condition, or this condition, and hit enter. And then you're going to hit equal, and then standard deviation, and go to the same data set. Hit enter. And then to get standard error, remember that is equal sign, your standard deviation, divided by SQRT of the number of samples, close parentheses, hit enter. Notice that first of all, notice the deviation. The oleic acid produced lipid droplets that have higher variability. So that would be something that you want to talk about in your results section, right, if this was your all samples. And then ethanol, right, this is our negative control, is the same thing, average. Go down and find all these samples, hit enter, equal standard deviation. I'm repeating this so you can see it a whole bunch of times. And then with this one, we're going to calculate standard error, which is divided by SQRT, double click. So that would be how you get that, and you can see that the negative control and lauric acid have very low deviations, which indicates that they're very precise probably in their sphere formation. Now, the one thing I didn't show you yet, and I apologize, is the number of lipid droplets. How do you get that? Well, you can go through and just kind of look at the number in each column, and then subtract 1 because we're starting actually at column 2. Or there's a function for that too to just make your life easier. You can go to equal sign and count and hit that function, and then go through here and highlight and hit enter, and what it does is it just counts the number of squares. It doesn't actually count the data, it just counts the number of squares. So that's a quick and easy way to do that particular data set. So hopefully from all this you gathered how to get data you need for your graph. So now the last thing we want to do is look at what's called a student's t-test. So what this is if the p-value, when you compare two groups, if the p-value is less than 0.05, then what that tells you is there is a significant difference between the two groups. And if the p-value, right, is equal to or greater than 0.05, there is no significant difference between the two groups. And so that's kind of like what you're going to be looking for here in your results. Now, how do you calculate p-value? So first of all, it's really important that you set up a table to keep yourself sane. So we want to compare the average area of lipid droplets for lauric acid with control. So however many groups you have, you want to convey that many comparisons. So you want to make sure you have all the comparisons worked out here. So we want to compare lauric acid condition to the negative control. We want to compare the positive control to the negative control. And then we want to compare the two the two fatty acids to each other. And so that gives us 3 different comparisons. So that's what we're going to do. So to do this, you hit equal sign and then t-test. All right, so t-test right here. And then it's going to ask you for the array 1, array 2, tails, and type. So this is what you're going to do. So we know our first condition is lauric, so we're going to highlight all those samples. And then you want to hit comma, all right? So then I'll show you, then it highlights everything there. So then you've highlighted that entire array, comma. Then the next array you're going to highlight. In this case, we're looking at control, which is ethanol. In this case, so we're going to go through that and highlight those, comma. and then ask you for tail distributions. And I'm just going to go ahead and tell you this without telling you all the details of while you can read about the differences between these different distributions. But this is a two-tailed distribution. All right, so 2, comma, and then it's asking you, is it a paired comparison, a two-sample equal variance, or a two-sample unequal variance? So in our particular instance, these comparisons are assuming equal variance. All right, so it's a two-sample equal variance, so it's a 2. So you've highlighted the first array of data that you're interested in, comma, highlight the second array of data that you're interested in, comma, 2, comma, 2, close parentheses, otherwise it might give you an error, and then hit enter, and then that is your p-value. And now you're looking at, is this less than 0.05? Absolutely. So then you can say in your results section, right, that there is, and you can even say p, if you wanted to do p-value less than 0.05. It's actually way less than that, so you can even go less than that if you can demonstrate even more significance there, if that's not even really a thing. But and then you would do the same thing for these other groups here, and you would determine the p-value just like we went through, right? So I'm going to show one more here. So what's it going to be is t-test. In this case, we're looking at oleic acid, right? Comma, and then you're going to highlight your ethanol here, comma, 2, comma, 2, enter, and there's that p-value, and you notice it is also less than 0.05, and so that both of these groups are significant. And then you would do the last one, okay? So after this, now we want to make a graph of this data. So I went ahead and did the last comparison here for the t-test, and it was significant as well. So now what we want to do is this graph. So what we want to graph, and I also relabeled these for simplicity, so I can just highlight this area here because we want to graph the average area of lipid droplets in arbitrary units. So we're going to highlight this data, and I've made the labels much more simple because this is actually going to appear in your graph. In your graph here, so I made just lauric acid, oleic acid, and control instead of having all those details. The details I'll put in the figure legend, but we want simplicity. So I'm just going to highlight those 6 rows here, and then we're going to create a table. So to do that, we're going to insert, and then here I just like a simple bar graph, and we're going to mess with this a lot here. Okay, so the first thing we want to do is delete the chart title. It just takes up space. We are going to create a title here on this axis over on this, but notice I don't have to do anything down here because it's all nicely labeled. I might change the formatting here because I don't like the gray formatting here, so I might go in that and modify that. And you can do a lot of things stylistically, that you can work with. A couple things I want to show you here are some things that you do want to do, right? So first thing is we want an axis title here. And we don't need one down here because it's already appropriately labeled with the conditions. We do want a vertical axis. So the title of your vertical axis for this particular sample here is going to be average area of lipid droplets in arbitrary units. All right, and you can even say, I like average, you can even say mean area. There's different ways that you can go about this. So, I've seen a lot of different ways. You can just use the word mean area of lipid droplets. All these are okay. So a lot of times you'll also see some other ways of labeling that, but this is perfectly fine for today. So then what we want to do is click on the bars. So you've highlighted all the bars here. And so the next thing you want to do is go and you want to add error bars. Now it's going to stock in some error bars that are totally not even purposeful. We want to go to more options. So error bars, more options. And then what we want to do is we're going to customize our error bars to the actual data. Specify values. All right, so clicking on specify values, it brings up the positive, or what that means is above, the negative, which means below the mean. So what we're going to do here is we're going to highlight. We're looking at standard error, and so we're going to put the standard error in here, and not the standard deviation. So we're going to highlight now, and because we use the same order, we made this table, it makes our lives easy. So all we have to do is highlight the 3 values here. All right, so here we have our 3 standard error values. We come down here, and then we're going to do the same thing for the negative side. And we're going to highlight the 3 values, and then hit OK. And what it's done is it's updated our graph. So now, where we had that weird nonsensical error bars, now we have error bars that match the standard error. In our figure legend, we would want to say, in the figure legend, this is a graph of the mean area of lipid droplets under conditions of lauric acid, oleic acid, and control. And the means are presented plus minus standard error. And so that way, someone who's reading this table knows that these error bars represent the standard error of the mean. And in fact, I would be more precise. Standard error of the mean is what it should be saying there. I would also, in this figure legend, include data like the number of lipid droplets in each sample set. So, and you could, even if they differ, you could put it here in this legend. I've seen people put it above, here, which I kind of don't like, but you can do that as well. So we're almost done here. A lot of times I don't like the color blue, and you might want to change that depending on the situation. And so you can double click and highlight only one of the bars of interest. So let's say we want to change the control here. We can change that color, right, pretty quickly here. And so we can change that that particular, color here. We can change that to white, if we like. So let's go ahead and do that. Okay, and I like to highlight the black there. So I kind of like that. And then you can also change the color of the error bars. I don't like gray. It doesn't show up very well. So you can see how I manipulated both the error bars and and also the actual box as well. So I'll do that over here again. So maybe with the oleic acid, because you have 2 different conditions, you can highlight the oleic acid. Maybe we want to make this so the fill here will make like a gray, light gray, and the border will make black, so that it kind of matches. And then notice the error bars, they all, I did them all black, but once you click on the error bar, you can change that. You can also change the type of error bar, but I really like the way this is looking now. The same thing over here. Maybe this one, this is our condition of interest, but I would probably, just for simplicity, and then it changed everything. So double click, and then maybe I would keep both of the acids, unknown kind of things the same. And so I kind of like this, but once again, you can kind of do whatever colors that you fit your color scheme for your website, probably to be best. But this is what I would call a finished product. I would maybe, if it was me, I'd probably come and change the font a little bit and the color of the font to match whatever color scheme's going on in my website. But this would be, essentially what you need. There's one more thing that I would do is that you don't have here, I kind of almost forgot, is you want to include the p-values in here. And so how do you do that? There's several ways that one can do that. If you only have 2 groups, right, if let's say you just had a negative control and an acid, you could just above the acid put something simple, a text box. You could put a simple asterisk. And then in the, and then something like that, right? And then in the figure legend, you would put asterisk equals p-value less than 0.05, right? And then the reader knows that, and then you would also say, in the figure legend, you would say a student t-test was performed and a p-value was obtained of 0.05. However, here in this case, we have multiple conditions. And so it may be a value, to actually tell us which conditions you're comparing. Now, since everything was significant, all right, so what we want to do is we want to compare multiple things. So let's say you want to compare, and we have three comparisons here, right? And they're all significantly different, so you could use the same setup here. I don't know why this has a box around it, but I can always change that later. So the last thing you want to do is when you're doing something like this, we want to insert a line. And usually, if I can find things, yeah, there's a line. And so what I do is you can start at the very top of and go to the bar that you're comparing. So we're comparing, in this case, lauric acid and oleic acid. And then I would change the outline. I'd like black because I'm doing more of a black and white. And then I would increase the weight to something similar to what we're seeing already in the graph. So size 1 seems to be appropriate. And that way you know that this particular p-value is, comparable here. If you don't like asterisks, you can actually put p-values 0. I've seen this where you can do p-value less than 0.05, and actually have that in the comparison there. And obviously, I get rid of this box here, but that would be something else that you could do there as well. So I don't like fills and I don't want lines there. So that just kind of clears that up a little bit, and then you could do the same thing. So then what I would do, see if it'll even let me copy and paste. It does. So when I'm doing oleic acid and control, that was another comparison, so I'd have that. And then the last comparison, right, is lauric acid to your negative control here. And so I'd make sure that's level. And then you can bring this here. And so that's now obvious which ones you're comparing. You can say they're all significant. There's different ways to showcase that. But overall, that's the graph. And then you want to make sure this graph gets copied onto your website effectively. And so you can just, save and manipulate the graph. You can copy this. And what I would do is I would copy this and I would make this into an image. Because sometimes, and there's different ways you want to do that, but I would save this multiple ways. So I would save it as a JPEG. I would save this as a TIFF. And you can do that a couple different ways with Paint or with other Photoshop programs. And then I would leave this and make sure you're saving all your data, right? And multiple places. The last thing you want to do is all this hard work and nothing shows up. So this is a rather long video, but it's because I showed you a lot of things to do here when you're calculating area and showcasing that in a graph. So hopefully you found this valuable.